No mai, haere mai. Welcome everyone. Thank you for attending this webinar today. Just to let you know that we are recording and this will be posted up on YouTube at a later date. Kia ora koutou, ko Rob Fagelin toku ingoa. I'm your host today and here to start the first Rangahau Rising Webinar for 2026. This is a monthly korero with early researchers, career researchers working on bioprotection research. Firstly, is apologies from Nick Waipara, who will be the usual host for these events, but he can't be here today. Before we get stuck into this and hear more from Nils, we'll open with the karakia. On the Bioprotection app, the Tukutuku, there's got a list of the karakias from Bioprotection, and I'll just read from that now. Kia hora te marino, kia whakapapa pounamu te moana, he huarahi mā tātou e te rangi nei, araha atu, araha mai, tātou e a tātou, hui e taiki e. Okay, thank you everyone for joining us today to hear more from Nils, who will soon tell us about bacteria and their enemies with insights from studying their microscopic warfare. My name is Rob Fagelin and I'm a Senior Research Fellow at University of Otago in Peter Finnan's group. And my interest field is also studying bacteria and their enemies, but with a focus on bacterial CRISPR-Cas systems and more recently translating these into different biotech tools. I'm also a member of Bioprotection Aotearoa. And just a quick little update for housekeeping. Please keep your mics muted during the presentation. We expect it to run about 45 minutes. Nils will present the first part and then we'll have a Q&A for the last part. So for this, please type your questions and comments into the chat window and I'll read them out at the end and for Nils to respond. And if these can't be answered, if we run out of time, we will endeavour to provide a written response afterwards. And as I said before, this has been recorded and presentations will be uploaded to YouTube and available on demand. And if you know someone who you think might be interested in today's session, please share it with them. Our webinars are for anyone and everyone. So it is my pleasure to introduce Nils Birkholz. I've known Nils for about 10 years now, since he first visited Peter Finnan's group in 2016 as part of his master's at the Technical University of Braunschweig. Close. During this, he published an excellent thesis, and his work contributed to a top paper in cell host and microbe. His love of Aotearoa, or maybe it was bacterial defense systems, led him to return to Otago shortly after this for his PhD in Peter's group. And he finished this in 2021, obtaining an exceptional doctoral thesis award and a stack full of outstanding papers, including the sole first author in a groundbreaking Nature article that demonstrated a transcription factor that operated at both the DNA and RNA levels to regulate expression of a defense system. Since then, he's had a postdoctoral fellow in Peter's group funded by Bioprotection Aotearoa, and also a health sciences career development fellow from University of Otago. And this year he's promoted to a research fellow and started his Marsden Fast Start grant as an emerging independent researcher. During this time, he has made an impressive resume with at least 13 papers, and these are mostly in outstanding journals. And also he's received a number of awards, including the 2025 Illumina Emerging Research Award at the Queenstown Research Week and the 2024 Young Scientist Award for the Asia Pacific Protein Association. So Nils is also a great communicator, no pressure, Nils, but also a big supporter of Bioprotection Aotearoa. And with that, I'll pass you over to Nils.
Thank you, Rob. I'm just going to start sharing my screen here. I was also thinking what you mentioned, that it was quite nice to get this introduction from Rob, because he was one of the first people that I actually met in the Finland lab back when I first moved here just over 10 years ago. And he's also been a very nice mentor, supervisor, and collaborator on many of the projects that I'm going to talk about here today. So it's quite a nice, quite a good fit. The work that I'm talking about here will also almost go back as far, and then I'm just going to move my way into the present, I guess, and give some glimpses of the future, I suppose. So yeah, we're in our lab are generally interested in bacteria and their enemies, of course. So what we're interested in is just summarized on this slide here. So one of these things is horizontal gene transfer and mobile genetic elements. So the transfer of DNA between bacterial cells or from the environment into a bacterial cell as carried by these mobile genetic elements. And specifically, in our lab, we are interested in the interactions between bacteria and bacteriophages. Bacteriophages being these viruses that specifically infect bacteria and therefore also, can be considered these mobile genetic elements. And more specifically, in this interaction between bacteria and phages, we are very interested in our lab in bacterial defense systems against these bacteriophages, because bacteria, of course, don't want to be killed by bacteriophages, so have evolved different mechanisms to protect themselves against infection. And phages, in turn, have evolved counter-defense mechanisms to circumvent these defenses. And those are aspects that we've looked at in our lab quite extensively. And so while we are, and me in particular, I guess, we are very interested in this fundamental research of bacteria and everything that they interact with in nature. This is also quite relevant for the general public, because these horizontal gene transfer and mobile genetic elements, for example, are great contributors massive contributors to antibiotic resistance and virulence spread. So much of the antibiotic resistance that we're seeing emerging is caused by this horizontal gene transfer between bacteria, and many of the antibiotic resistance genes and virulence factors spread in this fashion. Bacteriophages, because of the spread of antibiotic resistance genes and the predominance of multi-drug resistant bacteria, bacteriophages have, especially in recent years, more and more been discussed as an alternative treatment against these antibiotic resistant bacteria, as an alternative treatment, because they are so highly specific and can specifically kill, for example, bacterial pathogens. That is important for health, but it's also applicable for plant pathogens in agriculture, for example, where plant pathogens are threatening harvests and orchards and so on. So that's also where these phage-based antimicrobials might come in handy. And past research from different labs around the world has also shown that this research into bacterial defense systems and counter defense mechanisms can yield really innovative biotech tools. Everyone has probably heard about Cas9 and restriction enzymes. These are all derived from bacterial defense systems. And so doing further research and identifying new bacterial defense systems can help us potentially find new tools like that. So there are many different reasons why we should be interested in these interactions of bacteria with phages and other mobile genetic elements. When I talk about bacteria-phage interactions, I always like to show this picture by David Goodsell, which is probably the best possible illustration of what happens when a phage infects a bacterium, and it's also based on actual structural data, so it's probably also the most realistic illustration of this process. So what happens is you get a bacteriophage that sits or attaches to the outside of the bacterial cell and then infects it by injecting its DNA into the cytoplasm of this bacterium. And then it uses its genetic information to make lots of copies of itself inside this bacterial cell, which will then eventually burst out and then can start the cycle from the beginning. So this here shows an E. coli cell. Probably most people have heard of that bacterium, and a phage called T4. In my work, I've mostly worked with a bacterium called Pectobacterium coratoverum, which is an important plant pathogen. As you can see here, it causes rot in potato plants, but also in and potato tubers, obviously, and also in many other crop plants. And I'm looking at the interactions of this bacterium with a phage called ZF40. And here's just another illustration of a phage or phage ZF40 infecting this bacteria cell. And early on in my PhD, I discovered a defense system that belongs to the bigger group of restriction modification or RM systems. And these systems work by using using two different enzymatic activities. We have a methyltransferase activity here, which attaches a methyl group to a certain short DNA sequence, and a restriction endonuclease that's basically a molecular scissor that will cut DNA if it lacks this methylation, but cannot cut it if it is protected by this methylation mark. So basically, the host DNA is generally protected through this methylation here, whereas foreign DNA will typically lack this methylation. And phages, for example, that lack this methylation will be degraded. And so this system that I discovered a few years ago is interesting for many, many reasons that I could probably talk about for 3 hours, but I will just keep myself very short and just show the organization of it here with the methyltransferase and the restriction endonuclease. And you can see the effect that this system has quite clearly in these pictures here, where we have a bacterial lawn of the host of this phage and tenfold dilutions of this phage spotted on top of this lawn. And if you use a methylated phage, it can infect this bacterium very well, because you can see the clear zones here, which then dilute out into individual plaques, which originate from single phage infections. But if you use a phage that doesn't have this methylation in its DNA, you don't get any infection at all. So that's how effective this system is. You can see this even clearer here, where we look at a strain that has a knockout, so a deletion of this restriction component here, where we now see a more than 1 million-fold increase in phage infectivity after this knockout of the restriction enzyme, because that is the component that targets this unmethylated DNA here. And so up to this point, everything was working very much as you would expect for an RM system in terms of distinguishing methylated and unmethylated DNA. Something surprising, however, came up when we looked at an additional strain here where we knocked out the entire RM system. So there was no change observed here between these two with unmethylated DNA. But when we spotted the phage with methylated DNA on top of this strain, we saw a small but very reproducible difference of about 10-fold between these two. And that was something that I almost ignored, and I'm very happy that I didn't. And based on this observation, we reasoned that in this delta RM strain here, these methylated phages were obviously being restricted by about 10-fold, and this appeared to depend on methyltransferase activity, because if the methyltransferase was present in these strains, we didn't see the same decrease. And so the hypothesis that we formed based on this observation was that this RM methylation by this methyltransferase appeared to suppress some sort of alternative defense that acted in the opposite way as this RM system. It didn't restrict the unmethylated phage, but it restricted the methylated phages by about 10-fold. So we actually then, through a lot of trial and error and different approaches, we ended up finding this other defense system, and which turned out to be a nuclease gene. And as the previous results showed, it was a nuclease that targeted methylated rather than unmethylated DNA. And when we looked at the promoter of this nuclease gene, we saw the start codon here, so the gene would go in this direction, and we see typical promoter elements like -10 and -35 sequences. But what we also found was this sequence here, which happens to be the methylation site that is being targeted by this RM system here, and which is also methylated by this methyltransferase. So we thought, okay, if we observe that methyltransferase activity silences this nuclease, maybe this is based on methylation at this site here. And we tested this through reporter assays where we basically fused this sequence here with a fluorophore and then measured expression of this fluorophore as a proxy of promoter activity. And we saw that in a strain that has the methylation, we get repression of this promoter pretty much to background levels, whereas we get expression from this promoter in an unmethylated or in a host that lacks the methylation where we expect this promoter to be active. And when we put a mutation in this sequence here that prevents this sequence from being methylated, then we see no significant difference between the two anymore. So this showed us that it was methylation at this site here in the promoter that was silencing, or that was the cause for silencing of this gene by this RM system. So the RM system methylates within the promoter of the nuclease gene, and this methylation leads to gene repression. And so what we have in this strain is a really sort of paradoxical scenario. So we have this RM system here with a methyltransferase and a restriction endonuclease. The methyltransferase methylates this sequence here specifically, and the restriction endonuclease is then prevented from cleaving this sequence. This restriction enzyme can cleave the unmethylated sequence, but then there's the other nuclease, which works in the opposite way. So we have cleavage of methylated DNA, but not unmethylated DNA. So these have opposing specificities and are therefore in conflict. So you would think they shouldn't be able to coexist in the same cell, because this methyltransferase would constantly be creating targets for this methylation-dependent nuclease. And then this previous result that I showed shows that this conflict is resolved because of the gene repression by the methyltransferase and the promoter methylation here. And so I find this really fascinating that this exists in a bacterial cell, and I'm wondering why it exists, and becomes even more interesting, in my opinion at least, when you consider that this RM system, or we have some good evidence that this RM system here seems to be part of something that is one of those mobile elements that I mentioned in the beginning. So it looks like this gene here was present in the cell first, and then at some point this RM system came in and somehow had to deal with this methylation-dependent nuclease, presumably through this promoter methylation. But we are looking at the moment into how this scenario might have emerged in the first place and how this RM system became established in the cell. And yeah, we're hoping to basically reconstruct this evolutionary scenario in our ongoing research. But the bulk of this work was published a few years ago and was done mostly during my PhD and shortly thereafter. And so restriction modification is one type of defense system that bacteria have and that I've done research in. Another type of system that many of you will have heard of, I guess, is CRISPR-Cas, which for the purposes of this talk, we can also just say it's basically a defense mechanism that degrades incoming phage DNA. You don't really need a lot of detail, even though the mode of action is a lot different from restriction modification and probably more complex. But all we need to know here is that phages to circumvent this CRISPR-Cas defense have evolved anti-CRISPR proteins, and there's a huge diversity of those, and they can inhibit this CRISPR-Cas immunity at various stages. And what you might think is that this anti-CRISPR protein can just be produced at a high amount when the phage infects and will inhibit CRISPR-Cas activity. But what we've found and what we've done a lot of research in is that this deployment of these anti-CRISPR proteins actually requires a very sophisticated mode of control. And we found that this is exerted by these anti-CRISPR associated proteins, or ACA proteins, which we often find encoded together with these anti-CRISPR genes. And what we found was, also using reporter assays not too different from the ones I showed before for the RM system, is that when you have a plasmid here that expresses ACA2 and another plasmid where you have the promoter of this anti-CRISPR operon linked up to an EYFP reporter, then you see that expression of this ACA gene or protein leads to a strong reduction in promoter activity. So obviously these ACA proteins, this one in particular, and apparently all the other ones that have been looked at, act as repressors of anti-CRISPR production. And we also looked biochemically at this interaction. So we took purified ACA protein, incubated it with purified DNA, and saw that, or specifically DNA with this sequence up here, and saw that there was a specific interaction between the two, as evidenced through this gel shift here. So based on that, we could form this fairly simple model here where we have an anti-CRISPR gene and an ACA gene, or ACA2 in our case, And we get transcription of this into an mRNA and translation into an ACR protein, and that can then go on to inhibit CRISPR, and the ACA protein, which can then go bind to the DNA here in the promoter and inhibit transcription. And that could have been the end of the story, but we had some additional findings that didn't quite make sense to us at the time, and so we kept digging a little bit further. And what we found through this following research was that this ACA protein not only binds to the DNA upstream of this operon here, which I just showed, but it also binds to an alternative binding site further downstream. And it doesn't bind this at the DNA level, but at the mRNA level. So first this needs to be transcribed, and then the ACA protein can bind to this. So here is the gel shift for this RNA protein interaction. We had a lot of collaborators contributing to this work, but one in particular, Max Wilkinson and also Derek Kimanius, together solved the cryo-EM structure of this protein dimer in complex with its RNA template, which falls into this sort of intricate shape here. And so we had to extend this previous model by an extra step where ACA2 also binds to this RNA here upstream of the operon, and then not only inhibits transcription, but also translation of this anti-CRISPR. And what's also remarkable about this is that ACA2 is a member of a fairly well-characterized family of proteins containing helix turn helix domains, which have been predominantly known as transcriptional regulators. So it was quite surprising to us to find that this not only binds to the DNA and does this transcriptional regulation, but also does this translational regulation here. And something that I didn't show any data for, but that we've described in the paper is that, well, you might wonder why we need this double regulation, and we did as well. And what we found, or what we hypothesize and have good evidence for, is that this dual regulation appears to be especially critical in, later on in the phage life cycle, when you have lots of copies of the phage genome in the cell that all get transcribed and make mRNA. And in this case, this seems to be the condition where this dual regulation seems to be especially critical. And so we've done all this work on phage defense, RM system, and anti-defense, anti-CRISPR regulation. And now I'm going to go over to slightly more bioprotection related work, I suppose. And before I go to this, I want to acknowledge two lovely students who've been visiting our lab. Both of them happen to be international. Sarah from Wageningen University in the Netherlands and Timothée from Paris, who have really done fantastic work in this project that I'm going to talk about for the rest of the talk. And so the basic idea of this research was that we wanted to look at a type of phage that is called a temperate phage, which has an alternative life cycle to what I've shown you before. So this is basically what we've seen in the beginning: phage infection, replication and assembly of this phage, and then eventually lysis and back to the start of this cycle. But these temperate phages, they can integrate their DNA into the host genome and remain there for quite a long time, or sometimes forever, and just stay there and replicate with the host cell. And this is basically how they keep existing until there is a certain trigger that leads to their induction and excision, and then they can go over into this lytic cycle here and make phage particles and burst out. And what has been known from a lot of research over years and decades is that these prophages can confer certain advantages to their host. So there have been papers that show that prophages can mediate defense against infection by other phages. It's also been found that prophages often actually contain phage defense systems themselves, which seems somewhat contradictory, but evolutionarily, it makes sense for a phage to provide these advantages to its hosts, and also to provide mechanisms to exclude other phages, because that keeps the host of this prophage alive and enables this prophage to keep existing in this host genome. So that's why it makes sense that we sometimes find antiphage defense systems in these phages. And that is what we wanted to do. We wanted to find new phage defense systems in prophages. And for this, we looked at a collection of Pectobacterium isolates that were sampled from various sites across New Zealand years ago. That was published in 2008 by researchers at Plant and Food. And we then got in contact with Plant and Food with Sandra Wisnowski, and she got these strains out of the culture collection and sent them to us. And we then went on to do some research with them. We didn't have genome sequences for most of these strains, so we couldn't just look at genome sequences and see if there was something there that looked like a defense system. And also, of course, we were looking for new systems, so we might not have found them either way. So I opted for a different approach. And what we did was we used a technique that has been known for quite a while, where you can grow your strain that you think might contain a prophage, and you can add a substance called mitomycin C or a different substance that induces DNA damage. And this will act as a trigger for this prophage to become excised and become active, basically. And when you do this with your bacterial culture, then these phages will burst out of the cells. And if you then spin down your culture and collect all the cells and cell debris at the bottom, then you might find these phages in the supernatant. And that it was Sarah did. She did this with all the strains that we had. She made these supernatants that we thought might contain induced prophages. And she then went on and spotted these phage lysates on lawns of all of these same 96 Pectobacterium strains. So for all of these strains, she made lawns, and then she spotted the same lysates onto those 96 strains. So altogether, she screened more than 9,000 different combinations of phages and of bacterial lawns and lysates that we thought might contain phages. And then this is an example of one of her plates, or like a section of one of these plates, where you have a lawn of one specific bacterial strain out of those 96 and spots of the different lysates. And what you see here is that these different lysates have different effects. Some of them lead to a complete clearance of this lawn, where you see no bacterial growth at all, and others don't do anything at all. And then there were some that do something in between. So they have some killing activity, not as strong as these ones here, but some effect on the growth of these bacteria. And she basically color-coded these different phenotypes. So yellow means complete clearance, and shades of blue mean sort of intermediate. And then she went and made this gigantic matrix of lysate and strain interactions. So here we have the 96 different lysates, and here we have the 96 target strains. And then she looked if she could see any patterns in this. And she saw, for example, that some of these lysates inhibited growth of a fairly large range of strains, as you can see here, and other lysates seem to lack any inhibitory activity, or at least mostly. And then there were some strains that were susceptible to multiple lysates and themselves produced lysates that were not very potent. And then there were some strains that produced lysates with broad killing activity, but themselves were not susceptible to many lysates. And that often included lysates that were otherwise very potent. So there were different patterns that seemed to emerge there. And so we wanted to take a closer look. And of course, we had to sort of narrow it down, because we couldn't keep working with all of these 90-something strains. So we somewhat randomly selected these two strains here to keep working on and investigate further. Somewhat randomly, because it wasn't entirely random. We chose these two because they have somewhat complementary killing activities. So we have lysates of EC90 and EC129 here. And what you can see here is, down here, for example, is that EC90 kills EC6, but EC129 does not. And EC129 works in the opposite way. And this is just another depiction of this, where this complementarity becomes quite clear. And one thing that Sarah noticed was that when in this previous experiment, she only ever spotted the undiluted lysates just to minimize the work at that step. But then for some interesting ones, she made dilutions and spotted these, as I showed at the very beginning with about the RM system stuff. And she never actually saw these plaques that we saw when we used our ZF40 phage, and which is again shown down here. At some point, you would expect these zones of complete killing to dilute into individual plaques originating from single phage infections. But she never saw this. She only ever saw a dilution of this killing activity. And we were a bit puzzled by this and wondering whether it was actually phages that we had in our supernatants or maybe something else. It might be that these bacteria produce some sort of antibiotic that has a killing activity, which would explain this dilution effect of this activity. And so we did some further research. For example, if it was a phage, we know that phage particles consist largely of protein and DNA. And so if we take these lysates and add proteinase, an enzyme that degrades protein, then we should be able to get rid of this killing activity. And that is actually what we saw once we chose the right incubation conditions. Here, if you add proteinase to either of these lysates, the killing activity disappears. So that seems to show that this killing agent that we have in this lysate appears to be protein, or at least protein seems to play a key role in this activity. And that could still apply to phages, so it doesn't rule out phages as the killing agent. And then we did an experiment where we took these lysates and we passed them through filters of different sizes. So this one here would only let through molecules smaller than 10 kilodaltons, and this one only molecules smaller than 100 kilodaltons. And both of these filters completely removed this killing activity. So whatever it was in these lysates that was causing this killing was apparently bigger than 100 kilodaltons, because without this filtration was the only condition where this killing activity was retained. And that, again, still applies to phages as well. So we still didn't really know what was going on. It could still be that there were phages in there, just that we couldn't see these plaques ever. And also, if you take these lysates and run them on an SDS phage gel, then you see a fairly defined pattern of proteins, of different proteins here, of different sizes, which also could still be true for a phage. They usually have similar patterns of proteins of different sizes because different proteins make up this phage particle. We didn't really know what was going on. And so we then thought, okay, if this particle that we have in our lysate here is so big, then we should be able to see whatever it is under an electron microscope potentially. And so Sarah took her lysates and went to the electron microscope and took a look. And what she would have expected if there was a phage or there were phages in there was, as a reminder, something like this here. And this picture is backed by popular demand from one person in the audience, which is a certain supervisor wearing a phage costume. And here is a more scientific depiction of this, where we have a capsid and a tail and tail fibers and a few other components. But basically, this is the rough image that you would expect in your electron microscope. But when Sarah took a look at her lysates, she only ever saw these tails here, these things that look like phage tails, but she never ever saw the capsids, the phage heads. And we therefore then thought this sounds a lot like something that we had heard of, but we had never really done any research in, which is called a phage tail-like bacteriocin or a tailocin. So what this is basically a weapon that bacteria use that is evolutionarily related to a phage and a phage tail, but it has been modified by a bacterium to produce and release into the environment and use to kill competing bacteria. And how this works is sort of seen in this image that Sarah also took, where you have a bacterial cell and a tailocin attaching to the outside. And this here is an animation that I found from a paper by a different group, which very nicely shows how these tailocins work in principle. So imagine this is the tailocin here sitting on the outside of a cell, and the cell would be about here. And then what happens is that this sheath here contracts and pushes the tail tube into the through the membrane of the target bacterium. And this puncturing then leads to the death of the target bacterium. And that therefore seemed to be, or it was a great candidate for, being the causative agent of all this killing that we had seen in those assays. And actually in her images, Sarah also often saw both forms of these tailocins, both contracted and uncontracted. So it was also cool to see that there. And So because we had now had this really good clue that talosins might be what caused this killing activity in our experiments, we then finally went and looked and got genome sequences of those strains and ran some prediction tools. So you can take a genome sequence and run it through a prophage prediction tool, and it will tell you where there are likely prophages encoded in this genome. And it will show you complete prophage regions, but also incomplete ones. And those were the ones that we were particularly interested in, because we weren't looking for complete prophages with the capsid, but for ones that were lacking the capsid and that only had the components needed for making the tail and the tail fibers. And Timothy had a closer look at this, and then he found these regions here in the genome that looked like they were great candidates for this, because they had that. They had tail components encoded in them and tail fibers and some other things, but they all lacked the capsid components. And so to test whether these were the regions that were responsible for this talosin production, Timothy went and made knockouts of these regions. He basically just went and deleted them from the genome to see whether the lysates resulting from those knockout strains still had the killing activity. And what was really nice to see is was how surprised and amazed Timothy was at how clear these results were that he got. He basically couldn't believe that it was so clear. But what we saw was that these wild-type lysates, while they were producing the killing activity as before, the knockout strain completely lacked this killing activity. And the same was true for the EC129 strain and its knockout derivative. So these knockout experiments then seem to very strongly suggest that these regions that Timothy had identified were what was responsible for the killing activity seen in SARS initial assays. And so based on this very accidental discovery in our Pectobacterium strains of these talosins, we basically had to extend our field of interest to also include this interbacterial competition via these talosin molecules. And you can then imagine that, similar to the use of phages as antimicrobials against specific bacterial strains, you can also imagine talosins being used for the same purpose, to target specific bacterial pathogens, both in health and agriculture. And obviously, either of these has pros and cons, and depending on who you ask, they will probably say the phages or the talosins are better. I will not take a side for now, but it does show that this research has a certain potential for applications in the real world as well. Finally, there are lots of people that have contributed to all these projects here, listed on the slide. I hope I didn't forget anyone. So I would like to thank everyone, and all of you as well, for listening to this. And if you have any questions, then fire away. Thank you.
Well, thanks, Nils. Yeah, thanks. Thanks, Nils. That was really nice work and exciting research. I am biased, I've known your work for a long time, but I really like your approach to using molecular and microbial techniques to sort of uncover how these systems work, and importantly, the questions that you're asking. And also, I think each block of work showed a really nice example of how important it is to look at the data, analyze it, and question things if they don't fit the model, because in all cases, you actually found really cool things.
Yeah, that was, I think, the nice thing about some of these projects, that some of them just sort of happened accidentally based on random findings that we just followed up on. So that was quite cool.
Yeah, serendipity, but you have to chase that as well. So it's really nice work. So now it's time for the Q&A. Please pop your questions into the comments, and I can read them. To help kickstart things, I've got a couple of questions here for you, Nils. So yeah, I really like the Taliesin work, and really interesting beasts. I was wondering how Taliesin producing bacteria avoid self-intoxication during their release.
Well, so generally they are specific for other bacteria, so they won't typically target the same strain that produces them. So what happens is that you have talosins being produced in the cell, and they basically lyse out of the cell similar to a phage, and then they are in the environment and can target closely related, but not the same bacteria.
Okay,
Yeah.
so a specific sort of attachment to.
Yeah, so it's similar to, how phages have tail fibers usually that define their specificity to a certain host strain. And so do talosins. They have tail fibers as well that define which target bacterium they attack. And
Okay.
people do experiments with that. They swap tail fibers between different talosins and then they can change their specificity through that. So it's also an interesting thing to do.
So that was my follow-up question because right at the end you mentioned that they can be used as microbioles. I was wondering if these could be engineered to be used as a precision microbiome for infections.
Yeah, no, people are looking into that, and we have sort of started or are in the process of trying that with our strains to see whether we can change the specificity just by swapping the tail fibers, for example. And yeah, we're trying that, and other people have done that before and successfully with phages as well as talosins.
Okay,
Yeah.
cool. Right, looking at the chat board, we've got a question from Leah Smith. What has been your favorite project and why?
Well, Leah knows the answer to that because I can't stop talking about it. Obviously, it's the RM story, which I'm still working on now, with a nice student who's also here. Abraham is working on that at the moment as well with me. Um, yeah, it has a lot of different facets that I'm interested in, and I would really like to understand how this entire system works with the interaction between these two systems. Yeah, and how it happened in the first place. Yeah.
Right, yeah, please write in your questions if you've got something to ask. There's another one from me is do you think the ability of anti-CRISPRs to regulate expression through both the DNA and RNA is widespread across many or all CRISPR systems?
Um, well, it's certain, or it seems like it might happen in not just our specific example, but others as well. It's also something we're looking at the moment, is to see whether other anti-CRISPRs might be regulated in the same fashion. So we do see RNA binding for other anti-CRISPR associated proteins as well. So we're looking specifically at this, or have looked specifically at this ACA2 family, but there are, I think, 13 different ACA families now, and some of them seem to, have a similar, or at least RNA binding, but how exactly it works and what this RNA binding does, we don't quite know yet, but it seems like it's not just our specific example that does that.
Okay, thanks. And I guess looking at your first block of work, as you say, there seems to be a paradox, and it seems a really complicated way for a defense system to work. That it's got a RM system for targeting unmethylated DNA, but then also a nuclease that targets methylated DNA.
Mm.
Could you comment on why it could be useful for a bacteria to maintain, two different systems that are, yin and yang of each other?
Yeah, we have some ideas. Um, well, initially when we found this, I sort of assumed that this nuclease was constantly silent and would just not do anything. But then the more I thought about this, the less sense it makes, because otherwise, if it was the case, then the bacterium would just acquire mutations in this gene and lose it over time. So there has to be some advantage to maintaining this system, this methylation targeting system next to the RM system. And one idea would be that, sometimes phages infect a cell with an RM system and they might escape this RM system by becoming methylated in time. And so if you get one of those escape phages with a methylated genome, it can basically just eliminate the entire population because it's now immune to that system. Well, assuming that there are no other defense systems there. And what I'm thinking, maybe having this and maintaining this additional methylation dependent nuclease might be sort of a strategy to target such escape phages that have evaded the the RM activity and have become methylated. And so that's also something that we're trying to look at now is this nuclease gene really constantly silent and inactive, or are there certain conditions, maybe certain subpopulations that express this gene and are then ready to strike if any methylated phages should emerge?
Okay, yeah, cool. Right. Well, I've got one more, Nils, before you let you disappear. So I guess stepping back a bit, and we have a few guests here that aren't from Bioprotection at Aotearoa. Perhaps you could comment on how Bioprotection Aotearoa has supported you as a early career researcher.
Um, Well, one thing that I was actually planning to mention at the end of my talk, but I might as well mention now, is that there has been really fantastic support from, for example, the communications people like Zoa and Anna, who are always very active in promoting the work of especially these ECRs, including myself. So that's certainly a way in which bioprotection has been super, supportive. Also, having lots of activities or planning activities on behalf of ECRs or the entire bioprotection fauna, basically like the Noho Marae events and the and other meetups where just opportunities basically for researchers from all these different fields to come together and discuss. So that's what first comes to mind when I think of how bioprotection has been very supportive.
Yeah, thanks. Yeah, I agree with that. There's a great sort of culture within BA of that support, and also it's really nice to hear the different points of view. Everyone's got different experiences and expertise.
Yeah.
So, yeah.
Yeah, I mean, sometimes I feel almost like our work obviously is very molecular, and it seems quite different from things that many other bioprotection people work on, which is more larger scale ecology often. So yeah, sometimes it feels a bit far
Yeah,
away
but
from
I guess
that.
it's quite nice that you've got that ecosystem of, we're part of this and we're helping others do these bits. So yeah, it's cool. All right.
But we have a question now from Sandra.
Oh, Sandra,
Since,
oh, thank you. Will talosins induce resistance?
so will they basically lead, or will bacteria develop resistance to them? Is that the question? I'm assuming. Um, yes, that can happen just as with phages. Bacteria can mutate the receptors that talosins use to attach. Yes,
Comparing
that can
to
happen.
bacteriophage, how do you see them in terms of resistance? Other bacteria uh, having resistance?
You mean comparing talosins and phages?
Yeah. Do you think they're better positioned in terms of creating resistance? Is that a question?
I think in that regard, they are probably quite similar because they use similar or often the same type of receptor. So bacteria will respond to that in similar ways, I expect.
Okay, thank you.
And yeah, just one more before we finish, Nils. Zoha has asked, in studying bacterial phage interactions, what do you think is the most important for us to understand? Big picture here.
Yeah, I think maybe the most important thing is that while this all seems very small and maybe therefore unimportant, it actually has a huge impact on our lives as well. Like phages and bacteria have really massive impact on ecology globally. They have a massive impact on health, on agriculture, on so many aspects of our lives, really. And I think if you want to take one thing away from this talk, then let it be that studying these very tiny microscopic things does have very crucial implications for much larger things in life.
Very good. Well, that's good place to end. So, thank you everyone who joined us today for Rangahau Rising. This is our first webinar for 2026. Please head over to the Bioprotection Aotearoa's website for more information on Nils's work that he's done with BA, and you'll find links to the webinar and other information publications. And he's got links in the chat too. And in April, we've got Dr. Elizabeth Elliott No, who will be here to share her postdoctoral research, with her title being Dairy Farming in Aotearoa, Feeding the World and Destroying the Planet. Check out the chat for the links to register for this. And before we head off, we'll close our webinar with a karakia, again from the Tukutuku app. Kia whai kāria te tapu, kia wātia ai te ara, kia turuki whakataha ai, kia turuki whakataha ai, hui e taiki e. Thank you, everyone.